Good morning, neighbor. This is JC on a bike. I'm biking into the University of Pittsburgh today. Another absolutely ridiculously warm and unusually sunny day for mid-September in Pennsylvania, as far as I know. Just getting better every year. My wife is on the phone with Norway, so I guess I don't have anybody to say goodbye to. I got my phone, I got my wallet, I got my IDs in my back pocket. I should be ready to go. So it's Thursday. 2 more days left in the week. Let's get this done. And yesterday or the day before, I tried to do a journal club for the undergrads in neuroscience, and the idea was, I hope that's good, the idea was, in the original idea, like when I originally started writing, my idea was to just give you an introduction to the paper, and then I sort of veered off the rails, and I wanted to have something up before the course was scheduled to discuss the paper, and there are a lot of pressures involved with, what the hell is that? Oh, it's a big turkey. A lot of pressures involved with, let's say, presenting or critiquing one of your fellow faculty members' papers, and I don't know what happened, but I just never got to explaining the paper. I read back and listen back to my journal club, and I'm not very happy with it. I mean, yeah, I understand that paper much better now, and I'm very happy. For myself, it was very useful. And for thinking about what the figures meant, for myself, it was very useful, but for your undergraduate course, I guess it wasn't very useful. So I'm going to try to do something just with this morning ride that can stand on its own, and then do an afternoon ride that can stand on its own. I'm going to see if I can start pulling that off, because I think that might be a better strategy. Keep me a little more productive and up to date. Somehow I feel like when I don't produce a video because of time or whatever, there's a missed opportunity, but then the next day there's also another missed opportunity if I don't film, and then I have 2 films to make, and I'm very quickly behind. And so for the infancy of this channel, while still trying to pull it off all by myself with a full-time job, my plan is to just make the videos I can and delete the videos I don't. Maybe I could save some of the footage, for rainy days or days when I don't ride, but it just doesn't seem really worth it because I get so much new footage every week. And the idea is to kind of keep it spontaneous, so, here we are. So once again, we get to the bottom of this hill, or we're coming down to the bottom of this hill, and I'll try and get on topic. So first I want to start with a shout out again to the journal club of the Pitt Undergraduate course. Thanks for watching, if anyone did, besides Quincy. Maybe Quincy watched it 21 times, that's also very nice of you. But for the purposes of this this journal club thing, I might try to add a little bit to the first paper that I didn't before, and that is what they tried to do in the clever use of the tools that they used. So in that paper, as you probably discussed on Wednesday, there was a question there that I think I nailed at least a little bit, so maybe there was one useful bit of information in that first hour, was that their main question was, compared to other cortical narrators where we know that there's quite a bit of topology in relation to the information that they code, there doesn't seem to be a lot of topology in the information coded in piriform cortex, so they thought they'd look in the microcircuit to see if there was any evidence of such an organization. And they found two aspects of the microcircuit that were sort of directionally biased rostral caudal. So in a nutshell, you can summarize that paper like that. I think if you're going to get a question on the exam, then the one question might be, what technique or what methodology did they use to stimulate? That's optogenetics. And in this case, they had an animal wherein genetically all the inhibitory neurons are expressing channel rhodopsin so that they can be activated. And that's how they measured the strength of inhibition, by just stimulating optogenetically labeled. And even optogenetics is something that I sort of took for granted that your class would know. And of course you must have discussed this on Wednesday, but I'll just rehash it again because I think it's so important. Taking an algal protein and modifying it a bit so that it can be expressed in a controlled manner inside of mammalian cells is a pretty monumentally great idea. There were probably 10,000 molecular biologists alive at the time that this occurred, or that this idea occurred around the world. There are probably 10,000 molecular biologists that were capable of, had the skill and requisite knowledge to do it. But the dudes who had the idea are the ones, that's just like PCR. PCR is kind of obvious if you know molecular biology, but the only reason why it's obvious now is because someone told you. But to see it for the beauty that it was, and then to hijack it and use it for all the things that we've used it for, PCR, that's brilliant. So taking that enzymatic reaction and hijacking it for all the things that we've hijacked it for. I'm not really sure how I got off on that thing. I was watching this guy behind me who definitely wanted to pass me, and I sort of lost my train of thought. Sorry about that. You see, he's going like 35 miles an hour in Millvale. But optogenetics is a pretty spectacular idea. That's what I was doing. In the sense of, there's lots of biologists who could have come up with this. We knew there were light-activated sodium channels involved in photosynthesis for a while. I won't attempt to guess because I don't really know how long we've known, but we've known for a while. But the creativity, the insight to say, well, I wonder if we could express that in a neuron and then use light to control neurons. The first time I heard that idea, I was like, oh my God, of course. All you need to do is know enough biology to say, oh, duh. But to think of it, to come up with the idea of using bacteria that can replicate DNA at different temperatures, to use their enzymes in a clever way with human DNA or mammalian DNA. You can just put it in a pot, put the temperature higher and lower and higher and lower, and voila! We got millions of copies of the same DNA molecule. If somebody explains that whole reaction to you in relative to how these enzymes work, number one, you should be shocked at the fidelity of the whole process. But number two, you should be awed at the simplicity of the idea and wonder if someone presented you with archaeobacteria that live in hot springs, would it ever occur to you, oh yeah, detour, sorry, would it ever occur to you to use them in combination? This is going to be kind of a pain, isn't it? Hey, but it's an alternate route. All you subscribers and everybody complaining in the comments about always taking the same way, hey, you got your alternate route here. And it's an excitingly bouncy one. So optogenetics is super cool, and it's still in its infancy in the sense of what we can do with it. And that's pretty cool. There's lots of more questions that need addressing using optogenetics that we just haven't gotten around to yet because there's so many low-hanging fruit. But essentially anybody and everybody that can employ it is employing it, because in a lot of ways there's just no cleaner way to stimulate while leaving the circuit intact, and that can work in vivo or in vitro a lot of times. And so that's the amazing leap forward that optogenetics has brought us to. It's hard to underestimate the contribution to our understanding of the brain that optogenetics will potentially contribute. But right now we're still, a lot of the stuff we're doing is more or less confirming what we already knew. And confirming what we already knew is very important, because when we do it with optogenetics, we get some much more convincing results that directly test many hypotheses that up until now we have not been able to directly test in an intact animal. But also, we get bonus experiments like this paper from your discussion on Wednesday. Bonus results where we can, even in vitro, greatly enhance the specificity and the fidelity of the question that we ask. Having optical control over all the neurons that express VGLUT1 is a pretty impressive, genetically specific feat that, let's say, 20 years ago would have seemed all but science fiction to anybody you proposed it to. And that's not a joke, that's literally the truth. It would have been, oh yeah, sure, when we have that technology, then cancer will be cured and we won't have any problems with anything anymore. We'll understand how the brain works, we'll be able to control everybody and everything. But of course it's not that simple. But at the time, I'm just saying that would have sounded like fantasy. Genetic targeting of light-activated sodium channels to specific populations of neurons in the mouse brain. I mean, just say that out loud 3 times and realize where you are. Even when I was in high school biology, those words would never have been tied together. It would have never made, it would have been science fiction. And so we're living in a very, very interesting time, there's no doubt. But a lot of the unhappiness that you see in your daily life on the road and the people on the street, it's the unhappiness that I would like to address with these videos too. I want to try and spread something that isn't yelling at each other, even though I'm one of the people on the road that's yelling. I didn't make a video yesterday because I yelled so many times on the street I couldn't bear to edit it all. It was like, just didn't want to do it anymore. But I saw like 6 people on their phone yesterday while I was riding, egregiously ignoring the drivers around them because they had a text to write. It's extraordinary, really. Hope he sees me. This truck is going right, so you gotta watch out. Oh, it's hot in the sun. So my guess is that you're going to see more optogenetics. You're certainly going to see more genetically targeted expression of proteins that alter the function of cells in order to test a hypothesis. You're certainly going to see that again. But I think optogenetics is becoming so prevalent in neurobiology that in this survey course you must see it again. And it's really such an extraordinary thing because it brings together so many different parts of your biology background. To already understand the concept that we have sodium channels which are activated by light, sodium pumps and proton pumps that are activated by light. And we can take these different plant proteins and alter them so that we can use light to move anions or cations or even ATP in and out of cells in the direction we want to whenever we shine the light on them. Now, to me, if I still explain it like that, it sounds extraordinary. Yet today, if I'm lucky, I think I have a mouse downstairs where an experiment similar to the one in that paper is possible to do today when I teach Shelby. So I'm actually quite nervous and excited when I go in on days like this because I really like to teach people how to patch. But I also feel like it's a bit of a performance. You want to hit a home run on the first day. And I feel like Shelby's a good student. We need to catch her if we can. She's really interested in motor systems and thinks patch clamp is something that she might at all enjoy, and we definitely need to try and get her in our lab. I feel like students are the heart of it. Not too many of them, but the right ones, because they're the ones that come through the lab and keep everything fresh. I mean, if it was up to me to find the novelty every day, I would have lost the heart a long time ago. I'm still disgruntled, but it's days like this I enjoy a lot. Can't tell what he's doing. It looks like he's going to go straight. So this paper has only patch clamp in it. And patch clamp physiology is something I've been doing for longer than I care to reckon. But it's not boring enough so that I could do it for that long. I guess that's the way to look at it. I think after you do something for that long, pretty near anything will be boring, but I still enjoy it. I still enjoy the idea of being able to make measurements that when quantified can contribute something to our understanding of the brain. It's just that sometimes I'm convinced that there are people employing these techniques and these methodologies without really understanding the limitations of them and without really knowing what they're doing. And therefore there's a good amount of patch clamp research out there, I think, that you can now, seriously, dude, come on, man. Are you kidding me? That was like the 5th car. That's amazing. You see what I mean? This is only in Pittsburgh. I didn't even run that light. I went when it was green. So optogenetics is something nice, and I'm sure that's why part of the reason they chose to start with that paper, because really that paper is way out of your league. And when I listened back to my 1.5 hour long diatribe about that paper, I realized that most of that was nitpicking out of your league. You know, I was asking questions that would be appropriate for a reviewer who was reviewing the paper, not for students who were just trying to get something from it. And I missed sort of an opportunity. To better explain to you, you know, what can be accomplished in vitro. And that's what that paper is a really great example of. It's a paper where they use in vitro techniques, so that's no live animals at all. And these are animals that their genetic manipulation is expressed from birth, so there's no, you know, you don't have to worry about surgeries and this kind of crap. The animal just gets put to sleep. We deli slice the brain, and we make recordings from the isolated microcircuits that we make or find in these brain slices. And when they make recordings from different parts of the anterior parietal cortex, or anterior piriform cortex, they notice a rostral-caudal gradient to the inhibition to certain types of cells. And the fact that it's certain types of cells does give them, it's a real nice observation. If it was in all cells, you might say it might be more a bias of the slicing of the slices. You know, maybe you could cut a different orientation. And I think I mentioned that in the first video. That would be one possible, you know, nitpicky suggestion would be you should do another slice orientation of this brain region and see if your prediction that it's a rostral-caudal gradient holds up. Because if it's a rostral-caudal gradient, then if you cut, say, perpendicular to that axis, then there should be no gradient, right? They should all be equal. Of course, there are other confounds with cutting slices in different orientations, but it's just helping you to think about how scientific papers and scientific experiments can be improved, and how you can start to look for controls that maybe weren't done or could have been done, which would help better argue that's too big for you, too small for you, I mean, that would help them strengthen their conclusions, that would support their conclusions even more. And the controls really are a lot of times the best way that you know that the experiment is valid. And I guess that maybe sounds like a stupid statement, but think about it a minute and digest it a little bit. That the more control experiments there are, the more the experimental or the manipulated group matters. And so a lot of times in today's, you know, high-paced world of science publications, there's a rush really to get stuff out, and there's a pressure to make stuff sexier and to hype up whatever you're doing with the maximum hype. And maximum hype often leads to confusion or overstating the results. And so it has progressively gotten worse over the years that I've been in science, and so therefore the onus is really on us in the field to read papers and to distill what the papers really mean. What does the data really mean? What are the limitations of the data? And I think that's the sort of message I was trying to convey, although I did it very clumsily, is that you want to try and pursue a level of mastery where you can then think about the data and ask yourself, what other control experiments might be possible that would convince you even more that their data was correct. And I think that's a really wonderful question to ask yourself all the time. It can be a question to ask yourself in order to test your mastery, because the more of those questions you can potentially come up with, I think the better you're reading the paper, because there are always control experiments, even if they're dumb ones, right? Even if they're ones that technically have been done by somebody else. Remember, this is a different laboratory. So any controls that were done in another laboratory don't necessarily mean that if they were repeated, those controls would turn out the same again. And so, again, this is the pressure of modern research, right? You never want to replicate anyone's experiments, and you don't want to publish negative results. But negative results are often the control experiments for the big show. And so, what negative results here are missing? You know, those are the kind of questions that you want to train yourself to ask regularly about a paper.